when we think about microbiology, we think more about bacteriology and virology, don't we? As being, especially as far as being medically or clinically significant. But fungal infections really don't get the all of the attention that perhaps they should. And these statistics kind of bear that out, and these aren't even terribly recent. These are a few years old. So you see there the costs for healthcare with fungal diseases. And then I've just given you here really a couple of things that pertain to fungal infections. Yeast infections in the United States are a really significant problem with people who are seriously ill, especially in intensive care units. We see a lot of yeast septicemia, like candidiasis. That's a big concern for us. And then these other statistics here are on aspergillus, just one fungi that is clinically significant. And that, of course, aspergillus is really an issue with people with mold allergies, and that also with underlying respiratory issues like asthma. All right, so again, those yeast, the septicemias we see in the hospital, especially in ICUs, occur a lot of times because those patients have central lines. And it's very easy because we have yeast on our skin. It's part of our normal microbiome for healthy people, but it's there. And so it's easy to get those lines contaminated sometimes. And then when patients are on antibiotics that kill, what do antibiotics kill? Bacteria. And they do not have an effect on yeast for the most part. And why do you think that might be? I'm sorry? They are, and you know the difference in yeast cells and bacteria, the big difference, one of the big differences. We're going to get to it in a minute. Yeasts and fungi are eukaryotic. They're not prokaryotic organisms. So the antibiotics that target prokaryotes, different things about those prokaryotic cells, are not going to be effective against yeast. So because the yeast are in our bodies already normally, and we give patients antibiotics, then we have issues with yeast infections. Now, these other things you see here and other reasons that fungal infections in general have become so much more significant in recent years is because we just have a much larger immunosuppressed population of patients. Think about all of the transplanting that we do now with organs. All of those patients have to be on therapy, continual therapy, to suppress their immune system so the organs won't be rejected. That gives fungi that normally would not cause a healthy person a problem a chance to come in and infect. And then we've got patients patients with autoimmune disorders like rheumatoid arthritis that are on agents. And you see these commercials on television all the time for like Humira. And there's a number of those drugs similar to Humira. They also suppress the immune system to try and help with the conditions like rheumatoid arthritis. But the downside is they're leaving you more vulnerable for fungal infections. And then, of course, when the HIV epidemic happened, or pandemic, for a long time, we didn't have really good treatments, and those patients were very susceptible to fungal infections. Well, outside of the hospital setting, we're seeing increases in community-acquired fungal infections due to those things that we see there. It seems like we have a lot of extreme weather now, a lot more hurricanes and tornadoes and that sort of thing. Fungi, as we're going to see in a moment, a lot of the molds, their normal environment is the soil. That's where they grow. That's where they get their nutrients there. They're pretty happy. They really, for the most part, don't need us as a host, with a few exceptions. So the problem is we're in their environment, and when we're doing things that stir up the soil or the dirt or the dust, then we're going to have a chance of coming into contact with more of those spores and be more apt to get an infection. And that last thing there about the antifungal agents, we see, just like we've seen with the overuse of antibiotics in agriculture, we see that also with antifungal agents. Causes the same sort of problem. Okay, so I've already mentioned fungi are eukaryotic, not prokaryotic like bacteria. So, and they have distinct, things that make them distinct from animals and plants. We're really kind of more closely related to fungi than any of the other kingdoms or divisions, though, which is kind of interesting. Since they're eukaryotic, we know they have a nucleus, we know they have membrane-bound organelles, and they divide by mitosis or meiosis. But they do have cell walls, unlike our cells. Fungi have rigid cell walls with a lot of chitin and glucan. Those are polysaccharides, and they help give those cell walls their rigidity. Because chitin and to some extent glucan are kind of unique to the fungi cell walls, those can be targets of antifungal drugs. So sometimes that's what an antifungal drug will target. And then the cell membranes are a little different than our cell membranes. They're a phospholipid bilayer, just like ours. But where our cell membranes contain some cholesterol molecules to kind of stabilize the cell membrane, fungi contain ergosterol. So that means that's another possible target for antifungal drugs. Okay, they're heterotrophic. They're not photosynthetic. They get their nutrients from organic compounds like we do. And to a large extent from the soil. You see there, they can secrete enzymes out of their cell wall to break up large molecules that are in the soil, so they can bring those in and absorb the nutrients, kind of similar to what bacteria do. And so they play a big role in nature, in the environment, because they can degrade things like cellulose and wood and anything that's decomposing or decaying, that they can release those organic compounds back into the soil for the plants. So really helps with that recycling of those things. And that term saprophytic means just that that's where they get their nutrients. And normally, a lot of saprophytic fungi aren't too pathogenic to us. So here's just a few more things that make them important. We talked about those first two there. Yeast especially can undergo fermentation, just like bacteria can. And so they're used in a lot of production of foods like bread. Be difficult to make bread without some yeast, and other things too there that are important to our food and beverage industry. Look at that next one, that's huge. A lot of antibiotics are made from fungi. The first antibiotic developed, penicillin, was made from the mold Penicillium. So they have antibacterial components that can be taken, and we've made several antibiotics from fungi. They're used as research tools in a lot of research, yeast and I'm sure some molds as well. And look at that statistic, only 30% of fungi. There are millions of species of fungi. And most of them, as I've said, are just, they don't need us for a host. So a lot of it is an accidental sort of scenario where we become infected. I think there's more of an issue with plant infection of fungi than animals or humans. Yes. Yeast, yes. The yeast cells, and we'll talk a little bit more about the differences in yeast and molds, but molds not so much. So here, we'll talk about that now. So yeasts are the unicellular shape of fungi when we talk about, and many of them do live on our skin and mucous membranes. Molds are where the ones that have the hyphae, and they are multicellular, and they're the ones that tend to be environmental. Now, you see that term there, dimorphic. Dimorphic fungi, that dimorphic, obviously, two shapes. They grow as a mold at room temperature, or out in the environment, they're going to be molds. But at body temperature, they convert to a yeast phase. These are the molds that tend to be the most pathogenic for us if we do become infected with them. We'll talk about some of those. All right, so the way they reproduce, that's another interesting feature.
No, it's on the ground.
Oh, I didn't know the mic fell off.
Oh, there it is. Okay. Oh, the thing. Okay, the thing stayed on.
Does it just slip in there? Okay. Is that helping? I can't really tell. Okay, okay. All right. So the other interesting thing about fungi are the way they reproduce. So they have an asexual reproduction form and a sexual. And the asexual process, interestingly enough, is what is more successful at putting the spores out into nature, helping them to make sure that they disperse into new habitats and keep going. Of course, they use mitosis for that process. It is almost always the form we see in the clinical laboratory. So those structures that form the asexual reproduction structures are what we look at the most to help us identify fungi in the lab. Well, let's start with yeast. They are going to reproduce by mitosis. And this budding process there that you see, there on the top picture, you've got the nucleus that is going to duplicate there and just send that duplicated chromosome into that budding area there. Where is your laser on here, Dr. Daft? Okay. So here you see that duplication, it's going up into that bud through the microtubule. And then once that is complete, that bud then can break off, and that's called the daughter cell. You notice that there's not an even division of cytoplasm. And when we look at it under the scope, it's very common to see like a mother and daughter cell still connected, undergoing that process of budding. That really helps us know we're looking at a yeast when we see that budding process. So when we identify yeast in the lab, just to let you know, we look at that. We look at how they grow on the augers. Then we look at them under the microscope. There are quite a few biochemical tests we can do, just like we're doing biochemical tests for bacteria. There are several that we can use for yeast as well. And then, if need be, they can also be DNA sequenced to get a really good identification. Yeast will grow on some of the augers that we use for bacteria in the lab. Here you see, this is blood agar that we've used, and when you have a yeast on there, well, this particular yeast is quite white, as you see there, and Candida albicans, tends to be, it forms, do you see those little extensions out from the colony? Those are really common with Candida albicans. We call them feet-like extensions, but there are a few other yeasts that can do that as well. This is how it looks under the microscope, especially when it's been grown on a special kind of agar. These strands that you see, that's not hyphae, it's pseudo-hyphae, what we call pseudo-hyphae, because it's really just an elongation of those yeast cells. And then you see those, the blastoconidia, the clusters of the yeast cells at the tips of those pseudo-hyphae. And the other feature that's really unique for Candida albicans are those terminal chlamydospores right there at the tip. They're double-walled and round, and they're really unique to Candida albicans. So we can identify it, which, by the way, is probably the most commonly This is one more yeast I wanted to mention, Cryptococcus neoformans, and I wanted to mention it because it can be very pathogenic for the immunosuppressed population. You see how different the color is there on that agar? It tends to be much more of a cream rather than a white colony. It's also very mucoid, wet, shiny, mucoid looking. And that is because it has a huge capsule. I don't know if you've studied about capsules yet in class, but they can be a way that bacteria or yeast, it helps them adhere to cells. So it's a virulence mechanism. And Cryptococcus neoformans has a huge capsule. So this is an India ink stain. You just put a little drop of the yeast in saline on a slide and then add that really dark dye, the India ink. And it's kind of a negative stain because the yeast aren't going to take it up. But we can see in the middle, the little circle is the actual yeast cell. And that halo, that big part around the little yeast cell is the capsule. So you can see how large it is. Cryptococcus tends to cause meningitis in patients that are immunosuppressed. When the AIDS epidemic first began, we saw a lot of this in AIDS patients. Well, let's talk about the molds now. They are not unicellular, they're multicellular, and their basic structural unit, kind of the body of a mold, is the hyphae. And they will increase in length in an apical way with mitotic division. A lot of fungi have what we call septations in their hyphae, or they're like walled off areas. And the more evolved or the higher fungi, have those. It's considered an advantage because if part of the hyphal strand should get damaged, then the parts that are on the other side of the septation, they may be able to survive. Now, as I said, the hyphae will grow in an apical direction, and it will, fungi are not motile, all right? They don't have flagella for the most part. And so when they can detect that there's a nutrient source and they want to get closer to that nutrient source, they can grow apically toward in the direction of that nutrient. So that's kind of how they get closer to their food. And that big mass of hyphae you see there, we refer to that as mycelium. When you have a lot of hyphae together, that's mycelium. If we grow a mold in the lab, we have it growing on some sort of agar that it likes, the part of the hyphae is going to grow down below the agar so that it can get to the nutrients, whatever we've put in that agar. And then the part there above the agar we call aerial hyphae sometimes. That's the part that's going to hold those reproductive structures. This would work out in the soil too. It'd be the same thing. Part of the fungi would extend down below the soil to get to the nutrients. I'm not going to talk too much about classification. Fungi are kind of in a state of flux as far as their classification. As DNA sequencing that's now available, they're working on sequencing all of the fungi to get to compare their sequences, to be able to classify them more accurately. In the past, before DNA sequencing, they were classified according to their sexual reproduction structures. Well, that's a bit of a problem because we haven't found all the sexual reproduction structures for fungi. As I mentioned in the lab, we often don't see those. So it's hard for us to classify according to that when we can't even see all of the structures. But those three that are starred there, that's where a lot of our pathogenic fungi are. And this is just showing you the different sexual spores of those three genera, or kingdoms rather. The ascospores for ascomycetes, basidiospores for the basidiomycetes, and the zygospores. But then look down at that deuteromycetes group. That's the group where we do not know about their reproduction structures. So they're not really classified in a really accurate way at the moment. But I was going to talk for just a second about the ascomycetes. That's probably the largest group of fungi that are pathogenic to us. And you see how the ascocarp there, that's again the sexual reproduction structure that we don't often see in the lab. And inside of that ascocarp, you see these sacs there with spores inside them. The sacs are the asci, and then the little ascospores are inside those. This is an example of one fungus that we do see the sexual form sometimes. And this was I took this picture because you can see on the left, or I'm sorry, well, on your right, the asexual structure. So here's a hyphae strand. This is a structure called the conidiophore that holds a vesicle, and the asexual spores come off of that. You can kind of see them there. Asexual spores are conidia. This is that's the structure we normally used for identification. But in this field, you could also see this structure over here is a cleistothecium, a kind of ascocarp. So for some reason, this fungi tends to produce both in more frequently than most fungi do. And that's why I kind of took a pencil eraser and pressed on my tape prep, and I got that Clastothecium to burst open. And the ascospores inside are red, which I think is kind of neat. All right, that's just kind of how we go about mold identification. So you might notice what's missing from this list is biochemical reactions. We don't have a lot of biochemical tests for molds. So the biggest part of the identification for molds is just looking at these other things, and the biggest, I would say the most significant thing is the microscopic morphology. So it can be somewhat subjective, but I think that's also kind of what makes it really interesting. Because you have to look at how quickly it grows. Some of the more pathogenic, the dimorphic fungi grow on media with antibiotics, others won't. And then the colony morphology can be really helpful. The source of the specimen can be helpful. I mean, if you've got a respiratory sample, you wouldn't be looking for something like ringworm, for instance, the organisms that cause that. So that can help you some. All right, if we talk about the different infections that they cause, we can group them this way. So we have some that cause more of a cutaneous infection. What do you think that's, what are we talking about there? The skin, right? Skin, hair, and nails. Subcutaneous, these infections tend to go a bit deeper into the dermis, into the connective tissue. And then systemic infections obviously are going to start somewhere, and then they're going to spread, usually through the bloodstream. So let's look at these. The cutaneous mycoses are fairly superficial. Tissue is not invaded. We think a lot about this one, dermatophytosis, the ringworm infections. They're, It started what? No, they don't start in the bloodstream. That's how they spread, the systemic ones. They're going to start in maybe a wound or the respiratory in the lungs, and then some of them have the ability to spread into the bloodstream and get to other organs. All right, so dermatophytes, 3 genuses there, 3 genera that cause those. And then we can also put Candida in this group because it can also cause infections in that cutaneous way, since they're on our skin and mucous membranes to start with. Although, as we mentioned earlier, Candida is also can become systemic and cause septicemia. So this is one of the genera that causes the ringworm infections, epidermophyton flocosum. And I wanted you to see that this is how it would look if we grow it in culture and then make a tape prep and look at it under the microscope. You can see that you have the hyphae there, and you can even see pretty clearly this is septated hyphae. You see those septations? And then the other thing that you're noticing are these, what we call sometimes call them macroconidia, because they are so large and they are segmented as well. And also, we call them allurioconidia, as you see there, because they form directly on that hyphae. That's how they form. So there's no other structure like a conidia for them. They just form on the hyphae. And there's a picture of what it does to our skin. All right, now the subcutaneous infections are a bit more serious. So usually the mold spores have to get down in through a cut, some sort of traumatic implantation to get them fairly far down in there. But they're normally things found in the soil. And some of them adapt well to our tissue and some don't. So, but there are some that can cause some serious problems for us. Here's one of them, Sporothrix shankii. And it looks so pretty, doesn't it? It's got the hyphae there that you see, the strand of hyphae, and it's also septated. This is a conidiaphore structure here. And then the conidia are forming at the tips of that structure, and also along the edge of the hyphae. Yes, Alanis. It's just a little division in the hyphae. We call it a cross wall, where there's just a wall there so that nutrients can cross it, but like I said, if that hyphae is damaged, then it can kind of break from the damaged part and continue to survive. So this fungi lives in the soil, and people who like to garden and they like to work with roses, this can cause something called rose gardener's disease, because the spores can be on the thorns. And if you get pricked pretty badly with a thorn, those spores can get down in your skin, and you can have something like this happen. Although I would think this is maybe, they let this go, I would think. It looks like maybe it started here, maybe, I'm not sure. And it will continue then to progress along that those lymphatic vessels. So you see how it's kind of moving up the arm from there. But clearly much worse than a sort of a ringworm infection. Now, Sporothrix is actually one of the dimorphic pathogens. It can, once it gets into the body, there's a yeast form, not that pretty mold form we just saw. So it's a bit more pathogenic than a lot of the subcutaneous infections. And it can become systemic. It can spread into the bloodstream and disseminate to other organs, although it doesn't happen as often as with the other dimorphic pathogens that we're going to mention. Here's another mold that causes subcutaneous mycoses. This is Phyllophora. And both of these, Sporothrix and Phyllophora, you can't tell it so much here, but they have a lot of melanin in their structure that gives them an extra advantage at evading phagocytes in our immune system. So that's another reason they may be able to cause more infection. And there again, you see the hyphae. I can get my thing to work. There it is. There's your hyphae strand. These are, we call these structures phyalids. Mycology kind of has its own language. And you see how, to me, they look almost like vases with that structure at the top, that's a collarette. And then the conidia are forming at the edge there of that collarette. So they kind of look like flowers in a vase. But in your body, it's not quite that pretty. This occurs mainly in the tropics and the subtropics, and a lot of areas where people don't have access to good health care. And this is obviously something they've let go for a long time, and it has continued to worsen there. We call those like cauliflower type lesions that have formed. So I think this probably would have remained localized had he gotten treatment, but because he probably did not get treatment, it has spread. All right, and then many fungi can become systemic with patients that are, as we talked about earlier, that are immunosuppressed. And there are some examples. We've mentioned some of them already. Zygomycetes can be really bad for immunosuppressed people, especially diabetics, where their diabetes is uncontrolled. These can get up into the sinuses and wrap around blood vessels, cutting off the blood supply into the brain. And when that tissue begins to get necrotic, then they can feed off that. And it's very fulminant and causes death very quickly. It's really horrible. Those last two terms, hylohypomycosis and phaeohypomycosis, hylo means clear. So those are fungi that don't have a lot of melanin. And then pheo means dark. Those tend to have more melanin. And there's just a comparison to show you that. When they have a lot of melanin, we call those domadiaceous fungi. So as you see, they can look really different with their microscopic morphology. All right, the dimorphics. That'll be our last group we'll talk about. So they are more pathogenic to us, probably because they have those two forms and they can easily adapt to our tissue. Most of them, other than the Sporothrix, we tend to come into contact with that by breathing in the spores. So the site of infection is the lungs. Many of them grow just in certain areas of the world, so we call them geographically restricted. And here's a list of those. Thankfully, there's not too many of them that have the capability to be dimorphic. And we see in our part of the world, in our part of the country, Histoplasma capsulatum and Blastomyces dermatitidis. Sporothrix is pretty much everywhere. It's pretty much all over the globe. Coccidioides tends to be in the Midwest and Mexico, South and Central America. Paracoccidioides is also mainly in Central and South America. Penicillium marneffii is in Southeast Asia. So there's the map for histo. We'll look at it for just a minute, since I think we have time. When does this class end? 8:50, okay. So you see there, we're pretty much in the hot zone for histoplasma. It grows quite well around here. Up and down that Ohio River Valley, but on over into Southeast Tennessee. And these are the structures for histoplasma. The mold phase is there on your left. It has septate hyphae. It has, and look at that, can you see that there are two kinds of conidia? It has the little microconidia that you see along in here, and then it's got these huge macroconidia, and you see the little extensions from those. We call those tuberculated macroconidia. They're very distinctive of histo. And then if it is grown, if it gets into the body, this is what we're going to see. And it's a very small yeast. It looks very similar to Candida species. Now, with all of the dimorphics, one thing that we have to do in the lab is we have to confirm the identification, either through converting it to the yeast phase in the lab or sending it for sequencing. Because as you see, there's another mold that looks quite similar to histo that is not, doesn't have a yeast phase and is not as pathogenic. That's Cephalomium. So it's always important that we have to confirm that identification for the dimorphics. All right, and that's just kind of what that says. conversion to the yeast phase is difficult for most of these molds. So we have quickly moved to the DNA testing because it's much quicker, and that says false positives can occur, but that really doesn't happen too often. The tests are pretty good. Uh-oh. So as we mentioned, most molds grow in the soil, and histo is no exception. It's in the soil, and we tend to find it a lot in areas where the soil has been enriched from histo standpoint with bad or bird droppings. So places like caves where there's a lot of bat droppings, it's a good place to find a lot of histoplasma. And anywhere where birds have been roosting, you will find histo there. Around chicken coops is a good place to find it. Old buildings where pigeons have roosted, all of those are good sources of histoplasma. So when something happens that soil is disturbed, where those spores are, and they're up in the air and we're breathing those in, that's how we come into contact with it. And then the mold is transformed, the spores, into yeast because of our body temperature. Those are some of the symptoms if people get really sick. Now, if we all went and had a chest x-ray today, if you grew up here, many of us would see like some scars where we've had histoplasma and never knew that we had a problem. We probably had a very mild illness. We might have thought it was just a little flu or even a bad cold. But when people are really sick and become really ill with it, they can get all of those symptoms. I think part of it depends on your immune system. So if you have a healthy immune system, you're not as likely to have as much of trouble with it. But if you get a really large volume, if you breathe in a lot of spores, a very concentrated amount, then even healthy people can become sick with this. All right, and that's just kind of what I said there. It is a disease of the reticuloendothelial system. So our macrophages do so much in trying to combat this. But they can actually become infected with it, and it can spread among the macrophages. So, and you know where we have those a lot in all of our tissues. But we see it spread to the skin, the mouth, the liver, the central nervous system, and the adrenal glands. There's an x-ray of someone with a pretty bad case of it. Look at his, how he got that. He was exposed to a large inoculum while cleaning the bird and bat guano from a bridge abutment. So he got a big dose of it, and he's got pleural effusions there. He's got fluid in his lungs, got pneumonia from histoplasma. This is from a patient who had AIDS, and this had already disseminated to other organs. This was in the peritoneum, where they found all of these budding yeast cells of histo. And here it is actually in the peripheral blood. You see all of the red blood cells, right? And right up at the top is a neutrophil. I don't know if you can see, but you might be able to make out the little yeast cells inside that neutrophil, where the neutrophils are trying to phagocytize and kill histo. You know, the problem with histo And really several other fungi they are able to withstand phagocytosis a good bit due to some of the enzymes that they are able to secrete. They can withstand that process, and that's what makes them more pathogenic for us. And there's another example of that dissemination where it has spread to the mouth. All right, and there again, it's just going over with you what happens. So the macrophages can be packed with yeast cells and fill those alveolar spaces in the lung. That just means that parenchymal means organ necrosis, that in this case, the lung starts happening. And then once those macrophages are overwhelmed with the yeast cells, you'll see granulomas form. So a granuloma is where you have macrophages that cluster together. T cells will come in to try and help, and they'll kind of try to wall off these organisms and keep them from getting out and being able to spread through the bloodstream. That is a big one there, right there. We used to think that once they were in those granulomas that they were not going to be able to really escape, that they would die in there, but they're finding out more now that maybe histo is something that can lie dormant the same way tuberculosis does and reactivate when your immune system can no longer keep those granulomas formed. All right, and that's what the Centers for Disease Control says we should do to try and minimize the risk, especially for patients in high risk groups. So people that are immunosuppressed need to be careful when they're around places where there are a lot of bird or bat droppings, because that soil could very easily be contaminated with histoplasma. They probably shouldn't be cleaning chicken coops or exploring caves. I knew someone in college who was a big spelunker, and he was healthy, but he became quite sick with histoplasmosis from crawling around on the cave floor where all of that histo tends to grow. And then tearing down old buildings, again, you're stirring all that soil up with the histo. So that's probably pretty good advice for people, especially that are immunosuppressed. All right, I think I've worn you all out enough. I'll stop there.
All right, thank you, Professor. Appreciate it.
You are welcome.